Hey, everybody, and welcome to a day in the life of a mycologist. One of the most emerging trends of study in the field of mycology is bacterial fungal interactions. We want to know how bacteria utilize fungal hyphae and mycelia in order to achieve a type of dispersal they normally wouldn't be able to, and to access ecological niches that are unaccessible to them without fungal hyphae. And to do this, we do a lot of time-lapse microscopy with fluorescently tagged bacteria and observe how they glide along the fungal hyphae. But these are just qualitative understandings of how these bacteria disperse and move. As mycologists, we also need to have quantitative understandings. And in order to do that, we assess on media how many colony forming units, or CFUs, make it through to the end in the system we're experimenting in the presence of fungus and in the absence of fungus. And because we want to better mimic the environments that these bacterial fungal pairings often occur in nature, we sometimes use combinations of multiple species of bacteria. So not just one bacteria and one fungus, but two bacteria and one fungus, or even multiple bacteria and one fungus. And that presents a problem for us because when we plate these bacteria to get the CFU counts and to determine whether or not the fungi help them in terms of dispersal, we now have to differentiate often morphologically similar bacterial species to get the CFU counts. When this happens, we use what's called selective media. And in this case, what you're looking at right here is a plating of two different types of bacteria, Staphylococcus and Listeria monocytogenes, and we want to determine which colonies are Listeria and which colonies are Staph. In order to do that, we plate them on a selective media that creates a black halo specifically around the Listeria colonies. The reason this happens is because Listeria makes an enzyme called esculinase, and in the media, we have esculin, and not surprisingly, esculinase hydrolyzes and cleaves esculin, and that hydrolyzed esculin then interacts with the ferric ions in the media, and that creates a black precipitate. And that black precipitate then identifies it definitively as a bacteria that can achieve esculin hydrolyzation. And because our system includes only Listeria and another species of bacteria that can't hydrolyze esculin, we know in fact that these are Listeria colonies.